and welcome to another episode of the Healing Lyme Summit 2.0. I'm your host, Dr. Mariah Hinchy, and today we're going to be discussing what is going on behind the lab door. So, we're going to talk about the global standards driving diagnostic accuracy when it comes to Lyme and other vector-borne disease testing. Here with us today to navigate through this pretty tough topic is Dr. Leona Gilbert. So, Dr. Gilbert is a docent of cell and molecular biology and the CEO of Tested Oi. This university spinoff company aims to get patients tested so that they can be treated and recover quicker. She has her doctorate in biotechnology and a vast experience in bioinnovation and biobusiness. In addition, Dr. Gilbert's several peer-reviewed publications also demonstrate a thorough investigation of how a common virus or bacterium can contribute to an autoimmune disease. Dr. Gilbert's research initiatives into complete diagnostic platforms and clinical profiling of patients for tick-borne and autoimmune diseases will allow a better understanding of how chronic conditions can be established with infectious pathogens as potential underlying causes. Welcome, Dr. Gilbert. Thank you so much for joining us.
Well, thank you for having me.
Absolutely. So tell our listeners a little bit more, like, how did you get involved in this? How did this become your career?
Well, I actually did a postdoctoral fellowship at Penn State University College of Medicine under Dr. Stanley Nades in the rheumatology department. And we noticed that actually the test kits that they were using for Streptococcus pyogenes actually was failing the patients because that bacteria was changing shape. And when I left my postdoctoral fellow and came back to Finland to start my own group, I had the opportunity and funding from an American philanthropist to actually investigate Borrelia and how it changes form. And then that really led the research into noticing that the test kits, that was being used for Lyme disease actually aren't recognizing that form. So in my research group with my PhD students and my research team, we really showed that there's a chronic form of Borrelia. It's not being used in test kits, and therefore we should do better for the patients. And that's what's kind of started me on this kind of idea that we need to do better for the patients because the test kits were failing the patients.
And I would have to say, after all of out of all of the bacteria out there, Borrelia has to be one of the most fascinating to study because of the way that it changes throughout, its life cycle. And you have multiple different forms of it in the body at one time.
Yeah, I agree with you. And also it's a low abundance bacteria. And we keep forgetting that that it is like you only need a few copies of this to cause havoc on the system. And compared to like with Streptococcus pyogenes, it's highly abundant, in the system. You can really detect it directly. But Borrelia is low abundance. It clears the bloodstream so quick. It finds its niche, it causes havoc, cause problems and long-term problems. And no doubt it's difficult to detect it and to treat it because these test kits are failing and it's a low abundance. So
low abundance and very, very slow growing.
yes, exactly. Exactly. Yeah.
Okay, so for our listeners at home, today we're going to be talking about the International Organization of Standards for Quality when it comes to testing. So this is a little bit different than our other testing talks that we've had where we've talked about, direct and indirect and how to read these tests. So we're actually going to be talking more about like the regulatory side of testing. So, Dr. Gilbert, let's start by talking about ISO and really how these various international organizations apply to, most of our listeners are here in the United States, and then break down CAP and CLIA and FDA cleared versus FDA approved. Let's just spend a couple minutes kind of like navigating through all of this language when it comes to, these various clearings or approvals or certifications that all of these labs carry.
Okay. So I will start globally speaking. So the International Organization for Standardization, these ISO standards are harmonized standards okay, that countries all over the world can adhere to, conform to, and should be accepted, okay, by other countries, and henceforth ISO, okay? And the two ISO standards that we use for testing are the ISO, okay, let's say 13485, which is for medical devices. So medical devices that are being manufactured in whatever country, okay, if they are accredited by this ISO standard, they can actually demonstrate that their medical device is validated both analytically, clinically, demonstrates clinical utility, has a risk assessment, follows the really great quality management system in its production, okay, and also making sure that it is in real time always being validated and performing as it should for the patient. So safety, efficacy, so how efficient it is. So the whole robustness of that test kit, how it's performing. So we should not see high amounts or percentages of false positives or false negatives, because under this ISO 13485, those characteristics should be minimal in this standard. And those people adhering to this ISO 13485 for manufacturing that medical device, which these diagnostic kits are then covered under this ISO standard, they are audited every year to maintain this quality, okay, to maintain this quality management system, and to really demonstrate the validation analytically, clinically, and clinical utility, okay. Then the other side, to the clinical laboratories that are performing test kits, okay, there's another ISO called the ISO 15189. And this is really for medical laboratories to make sure that they are conforming with the quality standards in performing the test kits, maintaining the test results, okay, and even sample preparations from the day one that their sample comes in, how it's treated and how it's used, and how the reporting is for the patient or for the clinician. So we have the medical lab, clinical lab under an ISO, and we have the test kit being manufactured under an ISO. Those two ISOs come together, verify the consistent and accurate Lyme testing that can be done globally.
Okay.
Now, CAP is stands for College of American Pathology. They verify, okay, and accredited laboratories, okay, clinical laboratories under the ISO 15189. Okay, so CAP accrediteds under ISO 15189. So if you are adhering to this ISO, in a sense, there's equivalence to what CAP will accept. This body that regulates the performance of clinical laboratories. Okay, and when I say performance of the clinical laboratories, it actually demonstrates, equality in the efficiency and the quality of performance amongst all the CAP accredited laboratories. And that really boils down to this ISO 15189. So in a sense, if a lab in Germany is CAP accredited, okay, they will have the same quality, robustness, equivalence as a CAP accredited laboratory in the United States,
Okay.
okay? And one level more then, if those two CAP accredited laboratories use the same test kit that is manufactured under the other ISO, the 13485 for the medical device, then their test results should be equivalent and they should accept those test results. This means fundamentally that if a test result that is using a validated test like one manufactured under 13485 and in a CAP approved, okay, accredited laboratory in Germany, if it should be accepted in the United States, okay,
Okay.
under the CAP accreditation. And I'm only using those two as an example, but there are many countries adhering to some agreements, like this International Laboratory Accreditation Cooperation Mutual Recognition Arrangement. It's an agreement. I know it's tongue-tying here, but
Mouthful.
yeah, it's an agreement that has many accreditation bodies from different countries signing to the equivalence of the standards. Okay, so they should legally accept test results from other accredited laboratories. Okay, and there's a few of these agreements out there, and United States does does sign to some of these agreements. And that really means really that test results coming out of an accredited lab in Germany should be accepted in these countries that have signed these agreements. But that's not what's happening with our Lyme disease patients, okay? And I constantly hear these are false positives. It was performed in an uncredited lab, or the test is invalidated. Well, actually, those are common wrong and misleading narratives that I constantly hear because in order for a test kit that is manufactured under 13485 to get that stamp of approval, okay, to be accepted, internationally, and also legally to sell it, okay, the test has to be validated, has to be analytically, so the performance has to be validated, and then the clinical validation, so it has to be clinically relevant. It has to show clinically that it does properly diagnose for Lyme disease, okay? And so it's validated, yes, at the manufacturer's level, but under the other ISO, the 15189, that lab is legally obligated, okay, to independently validate that test kit, okay, in their own lab. And in order to maintain those ISOs and also because FDA requires that only validated test kits, okay, come into those labs, but CAP demands based on the ISO 15189 that these test kits be independently validated before they're being used under that accredited lab. So it's easy for people to disregard false, our test results, okay? Because just by saying it, but they shouldn't, because legally speaking, a test kit cannot be sold, okay, under these accredited labs if they're not independently or analytically or clinically validated, okay? So that means that uh, by just a clinician disregarding test results and saying, well, that's a false positive, that's actually legally an inaccurate statement, okay, according to these standards and legally selling these test kits, okay, in the countries. Now we talked, I know I'm talking a little bit longer than the 2 minutes, but to get back to the FDA approved and FDA cleared test, so FDA clear test actually refers to equivalence. So one test is equivalent in its performance than the other test kit. Okay, that is cleared. Being approved means that a new test kit, okay, that doesn't have a predicate or doesn't have a comparison test kit comes into the market, and it has to undergo more assessments because there's no comparative test out there to compare it with. But cleared means it's just performing the same as all the other tests. Okay, that's out in the market. All right.
Okay.
Yeah.
And then what about CLIA?
Yeah, so CLIA, so this is the Clinical Laboratory Improvement Amendments. Okay, that's what it stands for. It is another a lower level than CAP accrediting body for clinical laboratories. They do not verify to ISO 15189. They don't, unfortunately. Okay, so there is not a demand as stringent as CAP, like as stringent as ISO 15189. And so like, for example, some people will say that CLIA doesn't demand this clinical validation. Okay, that may be so. That may be so, but it allows kind of like in-house test kits to be used, in-house test kits, okay? So which means that that clinical lab could develop their own test kit and use it in-house under their own roof, okay? And in order for them, minimum standard that they need to be accredited is this CLIA standard. So they demonstrate that that test kit is analytically validated, okay, in its robustness, in its accuracy, and its precision, in its sensitivity and specificity. These are all words to describe the performance of the test kit. It doesn't have that demand of clinical utility, okay, as the ISO 15189. So there's a clear distinction between CAP accreditation and CLIA accreditation, okay? CAP is adhering to the ISO standard, the 15189, that demands clinical validation and clinical utility, not only analytical, but also that CLIA demands, of course, the analytical validation. And of course, there you want to show some clinical validation, but the clinical utility demand is not there. So there's a higher level of accreditation, higher level of demand in the CAP accreditation because it follows the ISO 15189.
Okay. So
So
for our listeners, if they
yeah.
want to be sure that the laboratory that they're using is following kind of like the highest global standard,
Mhm.
that would be looking to see that they are CAP, is it called certified? Like CAP approved,
Accredited,
accredited.
accredited,
CAP,
accredited. Yeah.
so everyone's CAP accredited is what you
Y-
would be looking for. And in the
Yeah.
United States, most labs are CLIA,
Yeah. Yes.
correct? Like you
Yeah.
have to be CLIA.
Yes, in my understanding, yes. And that allows then for for that lab to bring on their in-house test kits called laboratory developed tests, LDTs. Okay, LDTs. And we call them in-house test kits here on this side of the world, but it's a laboratory developed test. You have to be CLIA approved, okay, or accredited in order to carry and or to use your LDTs. All right. Most most like 99.5% of all Lyme disease testing are LDTs in these specialty labs. Okay. And I'm not saying that the performance is less than CAP accredited. I'm not saying that at all, but I think we should be aware that there are quality management systems and global standards, okay, that influence, okay, the performance and how we should look at these test kits. Okay. It would be so difficult to bring a test result from a CLIA approved test into a different country and say accept this because it's not adhering to the 15189 standard. It's
Right.
a
It's
lot
not
easier.
adhering to the global standard.
Yes, yes, yes. It and but like legally it's not, okay? I'm not saying that they can't get cap approval, and I encourage that they do in this shift of what FDA is making the, these clinical laboratories go through with their laboratory developed tests, but it's important for patients to know that by adhering to global standards, their test results should be accepted, okay, across country or even can I dare to say state to state or
Right.
province to province, and that's the key thing that we need to, empower the patient to think this, that it's actually these are my test results. You cannot dismiss them, or health insurances to say, no, we were not going to cover this. Well, you should because legally this is, it's accredited, it's validated test kits. There is no reason legally to disregard these test results. And that's what my message here for this interview is to empower the patients that, this is the information that you can bring with you, to say, don't disregard my test result and don't say it's a false positive or, or a false negative. I'm like, really, there are limitations to these tests. Yes, we understand, but they should be minimized because of these standards. And we manufacturers don't want to give false results. And hopefully
I
they
mean,
don't.
I think everyone listening or most people understand what a terrible time most patients have leading up to their diagnosis of Lyme and other vector-borne diseases. A lot of times they have seen, multiple doctors. They've been told that they're crazy. It's all in their head. They're making it up. They've been misdiagnosed with chronic fatigue, fibromyalgia. I mean, some very serious diagnoses as well that are pretty grim. And then, to be able to properly work up a patient and test them and actually have them have that positive result for them to go off to say their neurologist or rheumatologist or oncologist even and have them say, oh, that test that's crap. That's not a standard test, though that's a false positive or all of the narratives that I think we clinicians have seen over and over and over again. And it's like that patient having like that glimmer of hope that they finally had a diagnosis that was, able to be treated to then just be dismissed. I mean, I've seen it happen over and over again, and it is just absolutely devastating. So I think that this talk here is so important because you're literally giving the listeners like the language and the understanding that they need to have to be able to take this back to, that practitioner and nicely educate them that they're wrong, and
Yeah.
what actually is reality. So in addition to that, talk about
Mhm.
what other benefits these global standards bring to testing and to our patients.
Yeah, I really think that by looking out for this, you're actually protecting the patient more, okay? Because you're using really validated test kits. Like now they can use those words. Yes, if the test was manufactured under 13485, it's validated, okay? So you're protecting the patient so that they will not get a misdiagnosis, okay? And they won't, like, they get the most accurate diagnosis possible. And if that test was then performed under, this ISO 15189 accreditation, okay, then you can feel comfortable that that lab is performing that test kit as accurately as possible, okay? And that's the whole point, that mitigates the risks for the patients, okay? And hopefully then the patients will be managed more directly and more positively and more accurately, because we're reducing the risk of misdiagnosis. So we have then ultimately less false negatives, less false positives, okay? And hopefully this will lead to a better kind of understanding of reliable diagnosis. Okay. And at the moment, I'm not dismissing any clinical lab that has CLIA accreditation, because I think that's a great start and it's needed for the laboratory to develop tests. But if we are going to compare, okay, between labs, we need to get to that ISO 158 now for these labs, and also use test kits that are manufactured under 13485. Okay. So because there's demands on performance there. Also, the whole point too is, it because of these harmonized standards, it makes the market approvals streamlined, so we can get into those markets faster. Like right now, the testing scheme in Australia is very poor, because a lot of those test kits, and I say this credibly, a lot of those test kits are using, just the Borrelia burgdorferi senso stricto strain. Okay, you can increase the sensitivity, okay, and parameters of diagnosis by just putting the other two strains, Apsally and Guarini, in there, okay, and use, test kits that have those three strains, okay, and that's the whole point, that we need to do better. So streamlining with these ISOs and streamlining the regulatory, approvals based on these ISO standards or these accreditation standards and or in these international agreements, it should allow us to get into those markets, faster for the patients. So that's good for the labs and it's good for the clinicians, because there's a lot of clinicians, in countries that don't have good testing algorithms, they're wanting better testing, better testing. Well, we have better tests. It's just we need those countries to, to honor these agreements, okay? Honor the ISOs, honor these, recognize and enforce these ISO standards, and so forth. So, and also the whole long-term benefit with these ISO standards and embracing them legally more so, and empowering, patients and clinicians to utilize this, it's actually really uh, having an influence on minimizing chronic Lyme disease patients, the whole chronicity of these patients and long-term consequences in these patients, because we'll have, the good diagnostics tests from the onset, okay? And that's our hope, in educating more on these ISO standards and these global standards. And I'm not dismissing local standards, because CAP is really a good local standard in the United States. But we have to really think about, what's the benefit of the patients, and making sure that the standards are embraced, okay, amongst different labs, so that we have fair, equitable treatment, diagnosis and treatment for these patients.
Right.
Yeah.
Okay, so in the United States, we have CAP that's looking at
Yeah.
the ISO 13485 as well as the ISO 15189. But outside of the United States, what organizations and laboratories also comply with both?
Yeah. So so normally these accreditation bodies like CAP,
Yep.
okay, uh, uh, there are national ones. Like in the United States, there's a handful of them, okay, and just like there's uh, like CLIA is one, CAP is one, UAF is one, IAS is one. Okay, there's different accreditation bodies, okay. In Finland, we only have one because we're a small country. It's called Finnas, okay, Finnish National Accreditation Service. UK is UCAS, UK Accreditation Services. In Germany, it's DOCS, okay. So in some countries, there's a handful of accrediting bodies, but in some countries, there's only one that stand out, like in here in Finland. So these accreditation bodies, like I said, are they're approved by the government, the overall government, okay, and they do the assessment of these clinical laboratories, okay, and they are the ones these notified or sorry these accrediting bodies like UCAS, CAP, CLIA, um, they then assess the clinical laboratories in their performance. And they are the ones that sign the international agreements, okay, that recognizes that they are conforming with the standards of ISO 15189, okay? So CAP really is for accrediting alongside that they're verifying according to ISO 15189 for the medical laboratories. Um, but we have other auditors, if you call it that, other auditors for the ISO 13485. Okay. And these auditors, like Lloyd's Register, LQRA is one auditor, BSI is another auditor for the medical device manufacturing, that standard. So some of these standards, like the ISO, the fundamental one, the ISO 9001, that standard is for management of offices. Okay. There's different auditors for those standards. Okay. But the ones that we are really interested, that we've been talking about in the last few minutes, are really the 15189 and the 13485. Those ones are top level notified bodies or top level accreditors, okay, like CAP and FINUS and DOCS, and they are actually approved by governments, okay, to be accrediting bodies, okay. And here in Europe too, because we're European Union, Finland is a part of it. We even have the EU looking at making sure that these governments are appointing good notified bodies that can then approve legally for this IVDR on our side, which is the in vitro diagnostic regulation on our side. Okay, because that's the law that we have to follow. What we have been talking about are just standards. Okay, but the law is okay. IVDR here in Europe, and FDA has their approval system for tests, for tests, for tests. Okay, so FDA approves or clears, okay, for tests, manufacturing tests, and then IVDR approves and clears, or sorry, approves for tests that can be sold in European Union. Okay, and they themselves have their own harmonized kind of agreements as well. Okay, and it boils down to also if the manufacturer of the medical device is following the 13485. So if you are accredited to 13485 ISO, then FDA will accept it. Your quality management, Health Canada will as well. I have to say TGA in the in Australia. Okay, and EU will accept it. So the standards are important even to get legally your test kit sold. Okay, so we talked about accrediting of laboratories, and that's CAP, and that's FINNAS and the Finland, UCAS, and United States. That's accrediting for the laboratory. But legally speaking, even before you can sell a test kit, it has to follow the laws. And here in Europe, it follows the 13485 standard, the ISO standard. And the FDA is coming more and more closer to verifying the ISO 13485 standard for its testing parameters as well. So yeah, we didn't even go to that legal aspect, but there are
Right.
legalities.
Maybe in another
Um,
episode.
yeah,
Uh,
exactly. Yeah.
okay, so for
Yep.
those who might still doubt the accuracy
Mhm.
of the Lyme disease test,
Yeah.
you know, beyond the FDA and all of this accreditation, like how
Mhm.
how do the two different ISOs, the 13485 and the 15189, I'm just saying them over and over and over again in case our
Yeah.
listeners are trying to catch them and jot them down so they can look
Yeah.
into it.
Yeah.
But how do these ISOs address that?
Yeah. So it, you have to think that these two international standards are demanding real-time uh, performance of what they're doing. Okay, like the clinical laboratory has constantly have to demonstrate that they're performing the test kit, okay, accurately and robustly. How?
Yeah,
Through
how?
audits, through audits, through audits. Okay, so the ISO 15189, they are audited every year. Okay, CAP may audit every 2 to 3 years. Okay, but it's immense kind of demand in the beginning to get your approval, and then they'll come back and audit you. The ISO 13485, you're audited every year to maintain that credibility, robustness of your test kit. So what it's audit is are you using your quality management system? So are you validating your test kit clinically and analytically? Are you constantly demonstrating that your test kit is doing as it should be doing? Okay. And you need to show this. So you have to do a post-marketing surveillance follow-up. It's called report. So basically it's showing that it has clinical utility. It has no, not no, but it has very few false positives and false negatives. You have to demonstrate that. It has very little cross-reactivity. Okay. And you have to demonstrate, okay, with data, okay, that it is doing what it's supposed to be doing. So it's not misdiagnosing, it's accurately diagnosing, okay? And there's a lot more to it, but the test performances and the safety and the efficacy is the most important thing in this ISO 13485, making sure that the patient is not at risk if your test kit does something wrong, okay? And that's why we do have accurate, like I hear all the time, oh, no, we don't have accurate tests. We have poor testing. The algorithm that's used, the modified two system is outdated. Okay. And there's data to support this. But the test kits, there are test kits that are very good. Okay. With great performance. Okay. Great accuracy, precision, sensitivity, specificity. But but we have to use them at the appropriate time, okay? And that's the whole point with serology tests. There are pitfalls with serology tests. In all serology tests,
Right.
it's not exceptional for
Right. It's
Lyme
not
disease
conventional
testing.
versus specialty. It's timing is
Yeah,
everything during certain
exactly.
times of the infection. Absolutely.
Yeah, yeah, and that's the thing that we keep saying because I hear it like all of these great groups, that support chronic Lyme disease patients saying that we have bad testing. No, we don't. We have good testing. We're just using the test wrong. We need to start using it accurately, and that's what I'm trying to say is that let's use these good tests that are validated, okay, that are performed under accredited labs. Let's use them on the patients and change the narrative because we do have good testing. We
Right.
do. Yeah.
So there's no test that's 100% ever, right?
No, not
So
no.
do you know how, like, what the efficacy is, like, for a lab that has the 13485 ISO? Like, what is the margin
Yeah.
of error that's allowed?
Well, I would say that if we can talk sensitivity specificity, if I can say that because that relates to the error, like
So
anything of
explain to our listeners,
yeah.
because we're not all medical professionals, what do
Okay.
those two words mean? Break that down and then continue, please.
Okay. But I will in one
Okay,
second, but I would just say to start this, I would say that how well the test is performing, okay, is what the parameters should be looked at in the context of the patient and the clinician. So is it specific enough? Is it seeing what it should? And it is sensitive enough to see the negative cases, okay? So sensitivity and again, uh, a little jack like here. uh, but making sure that you're picking out the Lyme disease patients over the non-Lyme disease patients, okay?
Yep.
And specific enough that you can differentiate between the negative, like people that are not sick compared to those people that are sick, okay?
Okay.
And and I know and I've seen this in conferences last year. They're saying that we only have a 60% sensitivity rate, okay? Sensitivity rate 60%, that means like 60% of the chance you'll catch the cases, okay, and 40% chance you're going to miss it, All right.
But that depends on what test you're using.
Yeah, I know, but they're using that in their presentation, but that data is from scientific articles that are like 15 years old, okay? You need to look at the instructions for use. The instructions for use for the test kit is their legal document that demonstrates the sensitivity and specificity. Those two parameters are the ones that you should be looking at. So that shows how accurate this test is. So if it has a sensitivity of 98%, there's a 2% chance that it will not pick up as it should. Okay,
Right.
and if it's in specificity, it means that it's differentiating the negative people compared to the positive people. Okay, so those are the two parameters that we should be looking at. So a sensitivity of 98, like I said, 2% that they're not catching the positive people. Same thing, 98% of specificity, they're not picking up the negative people, 2% they're not picking up. So those parameters are good. I would think that a sensitivity of 95 or 98 is good. Okay,
Right.
these legal authorities and accreditors will not allow, okay, a sensitivity here in Finland at least, what I for because we're audited every year, would not allow a poor performance test kit to be able to be sold. And what that is, well, in United States, it's this FDA approved or cleared tests. So the sensitivity and specificity, what is acceptable at the FDA level, you need to look at those cleared tests, okay? And right now, I think IGeneX said it was 70, their IFU says it's 75% is their sensitivity. Specificity, I didn't see that in their instructions for use. And FDA accepted that, okay? But I know here in Finland, that would not be 75% sensitive would not be acceptable, okay? And so we have a demand to make sure that our sensitivity and specificity is higher than 90%. That's our demand here, not only our company, okay, but it is definitely a demand that we want to keep. But we have a legal obligation to make sure that those sensitivity specificity numbers are actually in the instructions for use. So I encourage every patient and every clinician to demand this legal document, the instruction for use of the test kit, and look at those parameters because that's the parameter. It's not the scientific peer-reviewed publications that are being, it's in the instructions for use. That's the legal document. That's what FDA allows it to be disclosed. This is what the European Union allows us to disclose, and our FINAS here, our regulatory body here in Finland, that's the legal document. So that's what we should be looking at. Okay. So and then because this is never given out, this instructions for use. But if you ask for it, they legally have to give it to you. Okay, and this is empowering the patient to be more aware and the clinician to be accurately comparing the test kits. Okay, because the instructions for use will also tell other parameters of the test kit. Okay, stability. Okay, the clinical utility, the analytical utility, all the validation parameters should be on this instructions for use. It's a legal document. It has to be there. And
Right.
I don't know why we're not doing that.
I don't know either. I think
Yeah,
right now it's also important to bring back up the fact that your test is only testing for what it says it's testing for. So we cannot
exactly.
say that we're testing you for Lyme disease and have that encompass all of the various species of Borrelia, as well as those species of Borrelia that also can cause tick-borne relapsing fever, which gets lumped into the Lyme diagnosis if we're using a test that is only testing for Borrelia burgdorferi, right? So it's like,
Yes.
again, your test might say, that it is, a certain sensitivity and specificity. But if you're only testing for one species, you cannot then extrapolate that and say, I don't have Lyme because you haven't evaluated like the other, what are we up to now? 18 or 19 different testable species of Borrelia that
Mm,
cause quote unquote Lyme disease. And it certainly isn't going to test for all the species of Babesia or Bartonella or Anaplasma or Ehrlichia or Mycoplasma or Rocky Mountain spotted fever, Rickettsia, et cetera, et cetera, et cetera, right? So know what the test is actually looking for in addition to knowing, its various efficacy and accuracy ratings.
I agree. Exactly what you like with what you just said. Exactly. And that's what it encompasses, that what that test is testing for. Like the legal term is the intended use of your test kit, okay? So the scope of your test kit. Absolutely. It doesn't have all the species, as you just said, of Borrelia in there. I absolutely like support exactly what you said. So when I, as a medical device manufacturer, I am constantly asked, well, this test result on this test was positive, but you're negative on here. Why? You can't compare the two test kits unless they have the same okay proteins in that test kit, unless they're testing exactly the same species. You cannot do a comparison, and it is actually really damaging for the patient to do that. Or even not only like if you're talking serology tests, if you talk about indirect tests and a direct test and comparing them, you can't do that.
Right,
you
right,
can't
not
do
at
you can't
all.
compare microscopy to serology tests. You shouldn't compare PCR with serology. You cannot do that. And even in the serology world, I go back to my first point, even in the serology testing, um, the different tests, you can have the same person being tested over against like 5 different tests, and the correlation or how they compare. Okay, this is called like positive agreement or negative agreement, how they positively compare with positive samples, how they negatively compare against, like that ranges from 70 to 98%, depending on what is inside of the test. What are your proteins? What are you actually testing for? And,
And I think
and
that's such an important point.
yeah, because like having one Borrelia species in a test kit compared to the three, I mean, most prominent ones, like global ones, that's they're, they may not catch the other two species of Borrelia, so that's the whole point of why we need to do better. We do have these test kits that have the most global, Borrelia species in them. We do have those test kits that test for multiple tick-borne diseases. We do have this, but we should not, and we need to stop comparing these tests. And then I just want to bring up one other point about false negatives and false positives. We are seeing in these patients, chronic Lyme disease patients, seronegative patients, which means that they're not producing, antibodies, because their immune system is dysfunctional at some extent. Okay, and it could be many reasons. All right, and there are many hypotheses and many studies to demonstrate this, and that's why we have false negatives. Okay, and we have to understand that in those chronic patients this is occurring. Okay, not in a typical healthy human that has a working immune system that can elicit, like build up, can make antibodies. Okay, we won't see that in those people, but when we're dealing with chronic patients, we are definitely seeing this, and this is not exceptional to chronic Lyme. This can be seen in other chronic patients as well, that there are seronegativity in these diseases. And that's something that we need to publish more on, okay, to substantiate that. But that's something to keep in mind, that when we're having a negative result, it may not indicate that they're not suffering from Lyme disease. It's just that their immune system is compromised and they're not being able to, build up those antibodies.
Right. And
So
it makes sense because so many of, I mean, really, I come back to, for me, the fundamental, like, why does Lyme become chronic? How does it become
mhm,
chronic? How does it evade the immune system? Because it breaks the immune system. And so, in almost everybody, it imbalance it, to say the least. But then when you have patients who have, impaired B cell production and they literally cannot make antibodies against it, and then we come along with these indirect tests, and they're called indirect because that means they're not testing for the bacteria itself, but they're testing the immune response to the bacteria, i.e., the antibodies, right? And so, it's like it doesn't make sense. Like, why would we use a test like that? And it's like, well, when it comes to Lyme or Borrelia, because of the way that it hates being in the bloodstream and it gets out of the bloodstream and kind of goes to the extracellular matrix and the collagen so that it can eat, and evade the immune system more deeply, it's very, very hard to get a direct test which measures like the actual genetic information of the organism. So it's kind of like the best in a bad scenario, right, is looking at the
Mhm,
antibodies. But again, coming back to Lyme is a clinical diagnosis, and all of these tests can be helpful if they're done at the right time with
mhm.
the right methodology, looking for the right organisms. But again, it's like, you have to take all of that and bring it into the clinical picture and into that one person sitting in front of you, right? And work them up clinically to get your
I
diagnosis.
agree. Yeah, I agree. Our test kits that we make, okay, as a manufacturer, is only there to help and aid, okay, in this clinical diagnosis. The full picture of the patient should be taken into consideration. And I reiterate that in including also an immune fitness, okay, testing, because because that's the whole picture of the patient. And there are very standard tests for this and for the immune fitness and we tend to overlook the whole picture of the patient and only want yes no answers. I know that's easy to react with yes no answers, but we're not dealing with a yes no bacteria. We're dealing with a chronic bacteria or a complicated bacteria, okay?
And one
And
of the
that,
things that for the listeners, let me just interrupt
yeah.
for a sec. So, what Dr. Gilbert is talking about is you can have an assessment of your immunoglobulins. So, you can have like your total IgG, your total IgM, and also IgA and IgE. But I would say, when you're looking to see if you're making antibodies, in a normal response, looking at IgGs and IgMs would be important. And it's something that you can get done and is accurate at pretty much any laboratory. So that way, if you're within the normal range, then you can probably take your test results, your indirect test results, more seriously if it's done with a credible lab. Versus like, if you're very, very much on the low end with either of those, then you would want to look at that when you're looking at your test result. And again, not just looking at positive or negative, but looking at, depending on the lab, like Vibrant gives you an actual number. IGeneX will give you an indeterminate or a 1 plus, 2 plus, 3 plus, etc. And so if you have low IgG or borderline low IgG, and let's say, you get an IDN as your result, that IDN has to be taken extremely seriously. Right. And the ones where so, anyways, I think everyone understands what I'm saying, but I just wanted to kind of break that down for the listeners so they understand what
Yeah.
you're talking about.
Yeah, and I would like to add to that too that even standard blood counts like for your immune cells, okay, like CD4, CD8, CD57, CD56, like these are coming out anyways
Right.
if they're doing blood counts. So along with the antibodies, like those are just standard tests that are normally performed. So let's look at those biomarkers, okay? And there are publications to demonstrate the correlation in to chronic Lyme disease patients, that have lower, CD57s and so forth. So the point is that that all gives a picture of immune fitness, and we need to employ those tests to say, okay, guys, come on, this is anyways being done. Let's look at that and correlate that with the test results, and think cleverly that that an immune system isn't, uh, uh, a one fit all for everybody. It's everybody's individualized, okay? And the immune system can have flares and come down and we see that in patients too. They have good days, they have bad days, or they have good weeks and bad weeks. And most likely it's the immune system partakes in this. So that's the point. Yeah, just to reiterate what and support what you just said as well.
Well, thank you so much for joining us. I want to make sure everyone hears that you are having the first annual conference of chronic
Ah,
infection pathologies. This is going to be September 5th through 7th of 2025, so this year. And what is the website that people can go to learn more about your conference?
Yeah, it will be first because this is the first. So and we're a little slow right now getting that official website, but it will be on at our website on the company's website. So www.testedtztd.com. It'll be there first, and then we'll have our own SIP websites as soon as we can get that up and running. So, but thank you for mentioning that. Yeah, we're excited about it because it's chronic infection pathologies, and it's not only just tick-borne related, but it also, of course, can be COVID, can be other infections that are attributing to chronic diseases. And I think we're seeing a lot of similar traits amongst these patients that have chronic infectious diseases and pathology. So, and even the science is demonstrating the common traits as well. So
And I
we
was
were
going to say it's
really,
data driven. Like this is a
yeah,
scientific conference where, your tagline is show me the data. So
yeah,
that's the
exactly.
main focus.
But I want to reiterate that this conference is unique in itself, that we want to make sure that it's impactful for the three major stakeholders in our area, and that is the patient, okay, that is then the clinician or the practitioner, and that is the scientist. So we will have impact statements and we'll embrace all three stakeholders, because without the three, we feel that we cannot progress further into having workable, tangible action, points for the patient, for the practitioner, and for the scientist. And we're unique in the sense of this conference because we're embracing all three. Traditionally, we see only for scientists or only for clinicians, maybe clinicians and scientists together, or then we'll have patient-oriented conferences. But we are absolutely trying to embrace all three stakeholders, and that's what we want to push in this conference. That everybody that presents, everybody that comes out, hopefully will come and provide impact for all patients, scientists, and clinicians or practitioners. Yeah.
Wonderful. Thank you. Where
Yeah.
is that conference?
Actually, here. So here is Uvascula, Finland. Okay,
Great.
Uvascula, Finland, and it's a great time of the year because we will not have mosquitoes, but we will be able to see the northern lights, hopefully, on clear sky. Yeah, it's still berry season, so picking season in the forest. You can go and pick mushrooms as well, and so it's a great time of the year to be here in Uvascula. So it's basically 300 kilometers north of Helsinki, and it's a great place to experience. So I encourage you all to come. And abstracts will be open probably next week for speakers. So I'm encouraging you yourself to submit an abstract with the data, and we say show the data because we want to make sure that everything that we do is credible for the patient, credible for the scientist, credible for the clinician. And it doesn't have to be peer-reviewed data, it can be preliminary data. This is to encourage, PhD students, master's students, postdocs, early career, scientists, as well as medical students and so forth, okay? Or even just stakeholders, okay, that want to come and let's say they analyze the, surveys, patient surveys, or analyze policies, but they have some data to show. So the whole point is really show us the data and to make changes in treatment, diagnosis, science, and outcomes for patients, scientists, and clinicians. That's the point. So show us the
Awesome.
data.
Well,
Yeah.
thank you. One more time, tell our
Okay.
listeners how to contact you or learn more.
Yeah, so you can email me at leona.gilbert, so Leona.Gilbert, Gilbert, at TESDED, T-E-Z, or Z, TED.com. I'm a Canadian when I said Z.
Mhm.
Yeah, so you can email me at leona.gilbert at TESDED.com, or go to our website, www.tesded.com, so TEZTED.com.
So
That's how Z.
TEZTED. Okay,
Yes, yeah.
gotcha. All right. Well, thank you again for joining us.
Thank
And
you too for having me.
my pleasure. For all of our listeners at home, thank you for joining us. And I hope that this information is helpful to healing from Lyme. Take care. We'll see you at the next episode.